Hey Sally, how are you?
Doing great.
Excellent. Well, it's exciting to be back for another episode of Reflections on the History of Modeling and Theory. As a reminder to our viewers, this is a part of the Society of Modeling and Theory in Population Biology, SMTPB, which is now at smtpb.org. And as well as our podcast, the goals of the Society include providing a forum for discussing theoretical issues and results, strengthening the connection of practitioners to journals in our field, building this community through meetings, workshops, and like this, online events, and fostering the careers of modelers and theoreticians in population biology. Uh, our podcast is one of a series of informal conversations with leading figures who have helped to shape our field and who can now reflect upon the changes in modeling over their careers. With the technical wizardry of Kaylee Charlebois, a graduate student at UBC, Sally and I are delighted to welcome Elizabeth Thompson.
Dr. Thompson, Elizabeth, did her bachelor's and master's at Cambridge University, then a PhD with Dr. Edwards at Cambridge University in statistics. She then went on to do a postdoc at Stanford, becoming faculty at Cambridge early on in her career before moving to the Department of Statistics at the University of Washington in 1985, where she remained for the rest of her career. Her career spanned the not just exponential, but super exponential growth in
Right.
genomic data and the information it reveals about our history and relationships. She was a major developer of sound statistical methods that have really revolutionized our ability to infer ancestry, genetic associations, like departures from Hardy-Weinberg, hybridization, etc. And our algorithms have allowed efficient calculations of probability of observing data across pedigrees and inferring things like relationships. Her work also established Markov chain Monte Carlo as a powerful tool for inference from genomic data, which is really the powerhouse today of a lot of genomic inference. Thank you, Elizabeth, for joining us today. And, I was reading one of your papers, Elizabeth, earlier about how Edwards challenged the field to turn genetics from just predicting, oh, this was what would happen in the future with relationships among individuals to an inference, to looking backwards and figuring out what the genes you see today says about the past. And I think your career successfully achieved that challenge.
Before we talk with Elizabeth, I'll just outline some of the major awards and honors during her career. She's been elected as a member or fellow of multiple societies directly related to statistics, but also broader interdisciplinary societies, such as the American Academy of Arts and Sciences. She's a member of the National Academy of Sciences and was also elected as a fellow of the Royal Society. As well as those fellowships and memberships, Elizabeth has received multiple awards for her work. These include the inaugural Jerome Sachs Award for cross-disciplinary research, that's from the National Institute for Statistical Sciences, and also the Weldon Memorial Prize from the University of Oxford, which is across a broad range of fields, and in Elizabeth's case was given for contributions to biometric science. So Elizabeth, welcome. Thank you for joining us.
Thank you. Thank you for that wonderful introduction.
Thank you for being with us today. Elizabeth, one of the first questions we'd like to launch off with is when did you realize in your career that quantitative methods, statistics in particular, had an important role to play in understanding genetic data?
Well, I think it was almost the other way around because I came out of mathematics and statistics as an undergraduate, especially probability and stochastic processes. And I think I was attracted to genetics as a graduate student because its basic laws were phrased in terms of probabilities.
Mhm,
So
mhm.
coming into genetics, it was always what the probability models and the statistical approaches could be used to answer all those wonderful questions about relationships among populations or individuals. And so the questions came from the genetics, and it was sort of almost natural from my perspective that the answers would be using probability modeling and statistics.
So it was a natural bridge. Was it hard to get into genetics at first?
I had never taken a genetics course. There was, well, through my advisor, Anthony Edwards, who referred me to lots of literature and lots of
Yeah.
papers. And I think I was incredibly lucky that I started in statistical genetics in 1970. It was a much smaller subject then, so much easier to get into in terms of the people and the literature and so on.
Hmm.
At 1970, 1971, we just then had Crow and Kimura published. So one had the basic textbooks to learn the probability modeling and so on in the background. We were just starting to get real data. Morant's big compendium of blood type frequencies in populations that were big by those standards, not very small by today's standards. But we were starting to get real data, so one could start to think more statistically and not just probability modeling. And we were starting to get computers. We got the IBM 370, the vastly enhanced virtual memory capacity through semiconductors. We got that at Cambridge University in 1971. So it was just perfect timing. I was just so lucky.
Right.
So thinking about that, the size of the field, I guess, and also the size of the data sets in the early 70s, something you've witnessed and also played a key role in is the development of methods that can be applied to extremely large volumes of genomic data that we now have. And so did you envision that at the time, that both the size of the data sets and then the need for statistical modeling would increase to the extent they have since then?
No, I don't think anyone could have predicted that. I certainly couldn't have predicted that. It's much more step by step. As you get new types of data or new data sets, they become available. And as the computing technology advances as well, then you see that it becomes possible to answer questions that you couldn't previously. And as approaches evolve and the methods evolve with the subject. So again, I think I was very lucky at the point at which I started was just the point at which it was starting to grow and I could grow with it.
Yeah. And were there, it is hard to predict, what the next methods are that are going to be useful. So were there methods in hindsight that were either more successful than you might have expected or less successful, things that went by the wayside that you would have thought would have become really important?
In terms of the methods, not really. I think I've just been very lucky at the way things worked out.
Mhm.
But I think I was very slow to get into genetic linkage analysis.
Hmm.
I was fascinated. I worked with Mark Skolnick and Chris Canning's University at Utah, developing computing probabilities that they were using for linkage analysis. But I was just so fascinated by the large complex pedigrees and genetic
Right.
isolates and what we could infer from independently segregating loci that I really didn't think a lot about linkage analysis in
Hmm.
that early decade. And it wasn't until I moved to Seattle in the mid 80s, well, start of 86, that I basically had to catch up because at Seattle, the Gott had been there years earlier, and that was where linkage analysis had really started computing probabilities on pedigrees with Gotts 10 years earlier, more than 10 years earlier. So I had to catch up when I got to Seattle and started.
Hmm.
That was where I started to get really involved in genetic epidemiology. Um, and I got to know Anna Matulsky, a wonderful, wonderful person. And then a bit later, connected with my long-term colleague Ellen Wiseman when she moved to Seattle. I was slow on linkage analysis. It was getting ahead of me, but maybe.
Were you slow, or were the data so sparse that you really, for a while, could just consider unlinked loci?
Well, no, I think that was true when I started, but we started to have restriction for argument like polymorphisms. Certainly at Utah, that when I was there visiting, they were talking about having maps across the genome and being able to
Hmm.
use these to find genes and all the rest of it. And as I say, for a while I heard, but I listened maybe, but I didn't hear.
Mhm. You've mentioned a few people. Could you tell us about the people that really influenced your career, your mentors, and the insights they gave you?
Well, mentors. Definitely my mentors. My PhD advisor, Anthony Edwards, of course. But if I had to mention two people out of my postdoc year, and I did a postdoc at Stanford, it would be Jim Neal and Jim Crow.
Uh-huh.
I was at Stanford, but I had independent funding, so that enabled me to travel and meet a lot of people. I was very lucky. Again, I just feel I've been so lucky. That's basically what I feel about my career. Um, and in the spring of 1975, I went to Michigan at the invitation of Jim Neal for 2 months. And he had data and he had questions. And he always seemed to think that I had the capacity to come up with answers to his questions. So we had an off and on, very long term, over many years, not all the time, but over many years, we had collaboration. And I think a lot of my peers, the other students and postdocs at Michigan, found him quite intimidating. But I never found Jim intimidating. He was always he always seemed to have confidence in me. And I think that was confidence that probably at that stage, as a very naive postdoc, I probably needed to get.
Right. And you mentioned Jim Crow as well.
Yes, Jim Crow. So, and that was also that same spring, just one day. I got invited by Jim Crow to go over from Ann Arbor to Wisconsin to give a seminar. And I gave a lunchtime seminar to which Sewell Wright came and Charlie Cotterman came. And Charlie Cotterman was someone whose then unpublished PhD thesis had sort of inspired me a bit during my PhD. Anthony Edwards had a copy, and that was where identified sense among closely related individuals, that's where that came from. And so Sewell and Jim and Charlie were at the seminar. And then Sewell and Charlie didn't stop talking for the next 10 hours. It was simply an amazing day. Jim was also embarrassed and said, they sort of took over the day. But for me, that was simply wonderful. Just, spend the day with those three people until I got on the late night Greyhound bus, overnight bus back to Ann Arbor. And Jim Crow was also someone who was always very, very supportive over the years. And he was a wonderful, wonderful person too.
Really gracious.
Yes, yeah.
And so that one day stands out though in your memory. Yeah,
yes,
that's amazing.
It really does. I can still see it.
Wow.
It sounds like at least at that point, and with the mentors and the people you met, that making of connections with new people in the field was easy, or it worked out to be quite easy for you to kind of expand the network of people you were working with or interacting with.
It was, as I say, it was a much smaller world in those days, and I think it was easier. And everybody knew everybody. I actually had met Joe Felsenstein a bit earlier in Cambridge, but I first visited Seattle again during that postdoc year. Luca knew everybody, of course. It was just a very small world, and it was much easier, I think, to get to know the leaders in the field.
Interesting that you'd say that, because travel was so difficult and challenging, so it'd be harder to meet people.
Maybe. I seem to manage to do quite a bit of traveling in there.
I guess it was Greyhound rather than Zoom calls at that time, but that worked.
That's right. That's right.
Okay. So we often ask people this question, so I'm interested to hear what you think about it. You described how you were developing new methods based on the data and computational power as it developed. But we're wondering if you have a favorite memory of a kind of aha moment where something, a model or just a method or a theoretical approach helped you to figure out something that had been puzzling you for some time before.
I had a hard time with that question. I had three.
Great.
Three's okay. That's fine.
The first one, which was a huge one for me, was fairly early in my PhD, 1972, when I suddenly had an aha moment for an iterative algorithm for maximizing the likelihood that Anthony Edwards was using in his human evolutionary trees. It was, in fact, an EM algorithm, but this is 5 years before we had the, quote, official EM algorithm. In fact, there were many gene counting out there. Cedric Smith with his gene counting much earlier is actually an EM algorithm, of course, also. So we didn't have the name EM algorithm, and we didn't have the unity across different latent variable problems. But recognizing that I could maximize the complete data log likelihood to get an iterative algorithm to maximize that likelihood was was my first really big aha moment, I think, uh, and something that I felt that really came out of me rather than being advised or taught.
Right. Rather than treating the pedigree as a whole, being able to step back through it.
Well, so that's a bit later. This was population
Okay.
human evolutionary trees, variation in allele frequencies. But yes, that actually leads to the second sort of aha moment. You mentioned the computational methods that we developed with Utah, and again, I think the essence of that is far more due to Chris Cannings than to me.
Mm.
But the idea that one could step through pedigrees and get by using conditional independence, not just for simpler pedigrees, as Robert Elton, of course, had already done, but that one could do this whole thing on these large, complex, what we now call graphical networks. But again, this was before computing probabilities on graphs in that way. But I think what I realized, and was an aha moment for me, was that it wasn't just for computing. They were actually already thinking about linkage likelihoods, which, as I've said, I really wasn't. But I realized, hey, we can use this to compute ancestral likelihoods. We can use this to compute extinction probabilities. And I was much more excited about being able to use it to compute extinction probabilities and ancestral likelihoods. And that was another very much aha moment.
So cool.
And you want the third one as well? Or?
Yeah.
Yeah, let's have the third one as well.
Well, that was when it comes to MCMC, when we were starting to get into linkage, doing a lot of linkage and multi-locus linkage and genetic epidemiology. And when we did the very early MCMCs, we took the latent framework of genotypes that Robert Elson and that we'd used in our other computational algorithms. And it was the recognition around 1990, 91, so fairly soon after we'd started, that what you should use is the meiosis indicators. The 0-1 inheritance patterns that might call inheritance vectors, but thinking them as an array, not just as vectors, an array over the pedigree and an array across the loci, and thinking of those as the latent variables that underlie your genetic data. And that so much enhanced the ability of those MCMC methods to get estimates of those multi-locus linkage likelihoods and so on.
Interesting. It's interesting in math and statistics when you see a problem from a different angle. Like that seems so related, whether you're looking at genotypes or which alleles are inherited. But the math can simplify.
Yes, I mean, the problem with the genotypes and the alleles was that, because, particularly multi-allelic loci, the basic MCMC methods weren't irreducible. You couldn't do them, or you couldn't guarantee that you could do them. Whereas with inheritance vectors, since they are a priori independent over meioses and Markov dependent over loci, so a lot of those problems that we'd had with the early MCMC just went away.
Wow. That's a great aha moment.
It made a lot.
Do you have any looking back on it, any insight about where those aha moments came from? Or just thinking about it for long enough, or it's just hard to say?
I think it's thinking about it, thinking about the methods, realizing that what you want to do is to maximize a likelihood or to compute a likelihood. So I think the first and the third one, it was by thinking about it. How on earth are we going to solve this problem of maximizing the likelihood or computing this multimodus linkage likelihood? And the genotypes just aren't working. And what other latent variables? And Seattle then, I was surrounded by people doing MCMC. Julian Bizag was there in Seattle. And he was very enthusiastic about the idea of auxiliary variables. So rather than using them, if you like, the obvious latent variables, you could use other latent variables. And yes, and so in a way that maybe came from the environment that I was in and talking to him. The middle one, thinking about extinction probabilities and ancestral likelihoods, it was because that was the question I was really interested in. I was interested in relationships among individuals. I was interested in who was the originator of this allele in this pedigree,
Mhm.
and already interested in the idea of some alleles surviving and some alleles becoming extinct from founders in the population. So I
Goes
think
back to,
so there it was the genetic question that I wanted to answer that made me realize, hey, we could use this method to answer that question.
Yeah. Yeah.
so both thinking broadly and having lots of questions bubbling up, but then also talking with others around you to bring in new methods
Definitely.
and approaches. Neat. Do you have a favorite paper of yours that you wish was better known?
I had a hard time with that one as well, but I do have a paper, but it's not one that I think people should go and read because it's actually not a particularly great paper.
Ha.
I think it has a very important idea, and this was 1976. I think it's my first TPB paper, I think, called Population Correlation and Population Kinship. And it basically pointed out that what allelic similarity at the population can tell us about levels of IBD or relatedness is very highly variable. There's a huge variance and a lot of uncertainty.
Hmm.
So there was a lot of stuff going around that time. There was Newton Morton's bioassay of kinship and a lot of other similar approaches that were basically equating expected values of IBD under some population model with the observed correlations that you saw on the observed allelic similarities amongst populations. Equating expectations, some sort of rather basic method of moments. And the same thing is true with F statistics. A lot of people, when they're using F statistics, they are looking at allelic correlations, but they tend to interpret in terms of relatedness, which at some sort of expected level is correct.
Yeah. Yeah.
But the process is highly, highly variable. And the uncertainty about IBD that there is simply from looking at allelic similarity is huge, particularly at single loci, a little bit less so when you look at averages over the genome, but there's
Mhm.
still variances, and you really need to, even nowadays, I think you need to consider the variation and the uncertainty.
Right.
I mentioned the seminar. I must have been very naive at the seminar that I gave at Wisconsin, and I actually talked about the difference between IBD and population correlation with Sewell Wright sitting there in the audience. But Sewell Wright was very, very clear. If you actually go back and read the early Sewell Wright, he talked about IBD and he talked about allelic correlation, but he never confused the two. Whereas I think at that point in time, too many people were just not really distinguishing between the two. So I think it's still something I worry about. And I'm very happy that I had those concerns back in 1976. So for that reason, I really like that paper. Though, as I say, I don't think it's a wonderful paper that people should go and read.
Right, but it's a good cautionary note, maybe. So there's something in there that's maybe a general lesson. It's when there's something you can connect with data, people are tempted to do it without thinking about the caveats, or in this case, maybe large variance that could differentiate the two things. So yeah, okay, that's great. We'll include that in our YouTube blurb when we publish the podcast.
Okay, thank you.
Okay, so I have a question about thinking about students, and so students now. Do you have advice reflecting on your career about how to approach the interface of statistics and empirical data, particularly biological data?
I do think it's much harder now because the subject is so much huger and getting into a particular area of the subject. But I do think the questions have to come from the science. And so you have to be interested in the science and know what the questions you want to answer are, and then you can develop the methods to answer them. I could never have done research in mathematics or even mathematical statistics, not because I couldn't do the mathematics. I mean, I was quite good at doing mathematics exams, uh, but because I had no feeling for what the interesting questions were.
Mm.
Mhm.
And as soon as I got to thinking about genetics, the questions were just there. The questions were exciting and developing, and they just kept coming. And if you learn what are the questions, get interested in the questions, you want to answer the questions, then your statistical or mathematical training will give you the tools to try and answer them, or sometimes to say that they can't be answered, that there isn't the information in these data to answer that particular question, which I think is equally important. Again, it goes back to uncertainty and
Yeah.
variation.
Do you think you had the sort of the right intuition? It was just seemed different when you started thinking about biology and genetics and genetic data. The questions, I mean, did it seem kind of obvious to you what were those interesting questions?
No, I think initially they came from my advisors and from Anthony Edwards. He was
Yeah.
already doing those human evolutionary trees that he'd started with Luca Cavalli-Sforza, and his likelihood, which was actually not quite a likelihood because he included the latent variables as part of the likelihood, which was the problem. But his idea that what we want to do is to maximize the likelihood, and got me involved in trying to do that. That came from him. And from Jim Neal, the questions came from Jim Neal. And actually, the second things I did as my PhD thesis, the questions came again from Anthony Edwards. Can we estimate relatedness from genetic between individuals from genetic data? So I think initially the questions will come from advisors who are in the science, probably, and they get you interested in the questions. But the student has to find what questions they're interested in. You can't just take the questions from your advisor, right? You've
Yeah.
got to evolve and decide what it is that you want to answer to.
Because
It's always sorry, go ahead, Sally.
you need that curiosity to stick with it until you can find the solution.
Yeah.
Yes.
I was going to say exactly that. It's much harder to work on something if you're not, when it all becomes difficult, if you're not
Yeah.
passionate about that question yourself.
Yes. So yeah, you've got to have the curiosity. And I think it's science, and in the interface of science and mathematics, I think it's the science that one is curious about.
Hmm.
I'm sure there are some people who are curious about mathematics, but for me, it's the biological sciences and genetics that that gave the questions.
And that leads naturally to our last question. What piques your curiosity at the moment? What do you see as open questions in statistical genetics? And what would you recommend for theoreticians to tackle now and in the future?
I can't do that.
Hmm.
Well, just what piques your interest at the moment?
That's two different questions. On the first one, as to where things are headed, I
Yeah.
think the questions about the same about a particular phenotype, a particular disease, or a particular area of disease and genetic epidemiology, it's the same questions. What are the relevant genes? Where are they in the genome? And what do they do? And how do they do it? But of course, now it's at a much broader and deeper scale than a deeper level, broader scale than I ever dreamed of. Um, and I think I'm sort of out of that field in a way because I think that large scale sequencing data has really changed the field of statistical genetics. I'm sure there are interesting questions and there are going to be interesting methods, but in many ways, I think the large scale sequencing has really changed the field. Uh, and that's already true for the humans. It's not true yet for other species. And it's not true for endangered species, which is where I'm happily involved at the moment. But probably the same will follow for other species as well. So with regard to statistics, yes, the tools will constantly need developing. But I think the core statistical training is still the same. You're not interested in the p-value or the estimate, but it's how to assess variability, how to assess uncertainty, quantify uncertainty, calibrate measures of scientific evidence. That, for me, is what statistics can bring to any science, I think. So, as I say, I think I'm out of it as regards human statistical genetics, but I'm still very happily involved with endangered species and things I'm interested in there.
Great. I think there's a huge, so you talk about the massive amount of sequencing that's becoming hundreds of thousands of whole genomes. It's crazy.
Yeah, yeah.
But there's also on the computer side is a move maybe away from the exact statistical reasoning that you're talking about and towards deep learning methods. And do you think that how do you see them as living together, the machine learning?
I think statistics becomes even more important. We've got to learn to assess uncertainty in those methods. We've got to learn to assess variability in what comes out. We've got to learn to calibrate them, our measures of degree of belief or confidence or whatever you like to phrase it in those things. So I think statistics is actually, I don't know quite how statistics are going to do it, but again, I'm sometimes glad I'm retired. But I think statistical thinking in terms of variability and uncertainty is even more important now.
Thank you, Elizabeth. That was very insightful.
Thank you.
And if you have one of those recent papers on endangered species, send them our way and we will add them to the YouTube blurb. Thank you so much for all the work that you've done over your career.
Okay, thank you. Thank you so much.
And thanks for your time in joining us as well. We appreciate it.
Thank you very much. It's been fun. It's been fun. I enjoyed thinking about these things and then talking with you.
Bye.
Bye.